We're going to continue our discussion of proteins by talking about their function. So DNA and RNA may be famous in biology, but they're just the instruction manual. Proteins are the heavy lifters. They can perform a hugely diverse array of functions, from muscle movement to oxygen transport to immune functions. You can see a lot of them listed here in this little figure on the left. The one I want to talk about in a lot of detail, though, is enzymes. And enzymes are biological catalysts. A catalyst is anything that speeds up a reaction. And catalysts speed up reactions by lowering the activation energy. If you look at the graph on the right of the screen, you can see it's plotting energy versus the reaction progress. And you have a starting energy, that's the energy of the reactants, and you have an ending energy, that's the energy of the products. But in the middle, you can see this part where the energy goes up a whole lot. And that's because that part of the reaction is the transition state. And that's this transient or very temporary structure that's between the reactants and the products. So to get here, you need a whole lot of energy put in, and that amount of energy is called the activation energy. What an enzyme does is it stabilizes the transition state, and that means you don't have to expend as much energy to get there. So the activation energy is lower, it's easier, and the reaction goes faster. So enzymes are recyclable. They can be used over and over again as many times as you want, as long as the enzyme is still working. And here is the reaction that an enzyme goes through. So you start with the enzyme and the two reactants in this picture. And when you have an enzyme, the reactants are called the substrates. And the substrates, or single substrate, whichever one, they're going to go into the enzyme. And the enzyme is going to stabilize that transition state and turn these substrates into the product, which is shown in purple. And that product will leave the enzyme because the enzyme does not have high affinity for the product, and the reaction will start again with new substrates. I like to compare this reaction to a matchmaker. So the enzyme is like a matchmaker, and two substrates or two people are going to walk into this matchmaker's office and be like, hey, we don't know each other at all, we don't want anything to do with each other, can you match us up? And the enzyme, the matchmaker, is going to go, sure, I'll match you up. And the matchmaker will work with these substrate people until they fall in love, and the product is them as a couple. Now, once they're a couple, the matchmaker has no use for them anymore, so they will tell them, bye, get out of my matchmaking office. I need to see a new group of single people. And the new single people, the new substrates, are going to come in, and the matchmaker is going to do their job all over again. Now, there's a couple different ways that people have looked at how enzymes work with the substrate. Throughout history, people used to think that enzymes work by something called the lock and key model. Enzymes are very specific to one substrate. For example, maybe the enzyme only works with glucose. And so scientists thought, well, it must have a very exact shape that only fits the substrate. And they called this the lock and key model because it was exact, like fitting a lock and a key. But some other scientists later on thought this isn't practical because if the enzyme is rigid in the structure of the substrate, how is it going to be flexible in order to create the transition state in the product? So they came up with a new model, and this is called the induced fit model. This is shown on the right with my kind of Pac-Man looking enzyme and the substrate going into it. So you can see that without the substrate in there, the enzyme's got kind of a different shape, and only when the substrate binds does the enzyme fit around it and make that exact kind of lock and key formation. So that's the basics of enzymes. We're going to get into the kinetics in a much later video, but this is what you have to know about protein function, about enzymes. I hope it helped you.